All right. Hello, everyone. Those online, welcome to another GRiP-ing seminar. Uh, we are delighted to have Natalia de Miguel, all the way from Argentina, joining us remotely to give excellent talk about a parasite that we don't often hear about, Trichomonas vaginalis here in the GRiP-ing series. Just a couple of announcements. We don't have any GRiP-ing or RUMs for the next couple of weeks. Bad news or good news, depending on who you ask. But after that, we have an overload of two GRiP-ing seminars. So May 20th, we have James Hewitson from the University of York. And May 23rd, Friday, so that's Tuesday. And then Friday, we have Mark Carrington from the University of Cambridge joining us. So I will let Lucas do the introduction.
Hi, it's my pleasure to introduce Professor Natalia de Miguel to the seminar today. She got her biochemistry degree in 2002 from the University of, and then she did a PhD under the direction of Sergio Angel, working in small hit proteins in Toxoplasma gondii. After that, she moved to the University of California in Los Angeles to do a postdoc with Professor Patricia Johnson, and there is where she started working with Trichomonas vaginalis. In 2011, she returned to Argentina, and in 2013, she opened her own lab working in Trichomonas vaginalis as a PI in CONICET, the National Scientific and Technological Research Council of Argentina. Since then, she's been doing a lot of research in this parasite, and in 2019, she became the first woman director or vice director of INTECH CONICET. And since last year, she became the first woman director of this institution. And today she's going to present work about Trichomonas vaginalis and the extracellular vesicles and the role in communication. Thank you, Natalia, for joining us.
Okay, thank you, everyone. And special thanks to Lucas and Claire for the invitation. I think it's not common that you have a presentation from Argentina and from Trichomonas. So I think it's, thank you for that. So I'm going to be talking about the work that we have been doing in Trichomonas for a few years now. And I'm going to give first an introduction because I think Trichomonas is not a very common parasite. So this parasite causes the most common sexually transmitted disease worldwide. If we look at the numbers of the last report of the WHO, we can see that the number of Trichomonas infections surpasses the cases of the numbers of chlamydia, gonorrhea, and syphilis that probably are infections that you hear more about than Trichomonas. But this infection is more prevalent in men and women in more than any other infection. So it is considered the most common non-viral sexually transmitted disease. And different to the parasite that you work on, this parasite has only one host. It only infects men and women, and it causes an infection in the urogenital tract that can range from asymptomatic to highly inflammatory. In most of the cases in men, in 90% of the cases in men, the infection is asymptomatic, so the men are generally considered the spreaders of the disease. And in women, in 50% of the cases, the infection is asymptomatic as well. But when the infection causes symptoms in men, they're usually associated to irritation and inflammation, causing prostatitis and urethritis. And in women, it causes vaginitis and all the problems associated to inflammation and irritation. But as I told you before, the infection is generally asymptomatic. So in most of the cases, it's not treated and it can remain chronic. And if this happens, it can also be associated to infertility in men and women, increased risk of cervical cancer and prostate cancer, as well as increase of HIV transmission. So it's important to understand the pathogenesis of this parasite because not only for the infection that the parasite caused, if not as well for the long-term consequence that they have. If we talk about the parasite is an obligate extracellular parasite. So it needs to attach to the host cell to infect and maintain the infection. So the parasite, when it's a free-living parasite, it has an ovoid form, and when the parasite attaches to the host cell, it changes its shape and increasing the surface area and changing a lot of the surface. And this process is key for the establishment and maintenance of the infection. And if we talk about the strains of the parasite, the strains are highly variable. Here I'm showing you 26 different parasite strains attaching to prostate cells in gray and vaginal epithelial cells in white. And as you can see here, the attachment properties of the strains are very different. We can classify them as a poorly adherent strain, some strains that are almost non-attached to the host cells, and some of them that are highly adherent. And these correlate very well to the form that the parasite grows in the tube in the lab. We grow this parasite as a bacteria. They grow in a tube alone without the presence of host cells. And if we look at the adherent strains that grow like forming these clumps in cell culture, where the less adherent strains, they grow all individually. So in the lab, we have been working for several years trying to understand which are the factors and components that are important for the attachment processes and the pathogenesis properties of these different strains. And what we observe is that there is a huge difference in the surface, the cell membrane protrusions of the different strains. Here I'm showing you a parasite stained with WGA, and you can see that the highly adherent strains that are here had a lot of philopodia and cytonym on the surface. If we quantify the numbers of philopodia, you can see that the highly adherent strains that are here in blue has more philopodia than the poorly adherent strains. Philopodia are considered thin and short cell membrane protrusions that have a role in migration, in adhesion, sensing, and antigen presentation. But also they have a lot of cytonyms that are considered specialized type of philopodia that can reach up to 300 nanometers and have a role in sensing and signaling. So the surface of this parasite is completely different. And the surface and the cell membrane protrusions in the last years has emerged as a way of the cells to communicate with one another. So here I'm showing you a more detailed electron microscopy to see the surface, the difference between the two different strains. Here is the highly adherent strains where you can see the philopodia emerging from the cells that is completely different to the one that is less adherent. And the same happened here in a video microscopy that I'm sure you are looking at the parasite fighting to each other through the flagella because they move very fast and they are very funny. They are very cool to grow. But I also want you to look at the numbers of protrusion that this cell has on the surface. It's huge and it's completely different. So we thought that they might have a role in the process of attachment. And as I told you before, the parasite, the highly adherent strains, they grow forming these clumps that are generally associated to the attachment to host cells, whereas the less adherent, they don't form these clumps. And if we look inside the clumps, what we observe is that by electron microscopy, it looks like the clumps, the cells are attached to each other inside the clumps through the formation of these long, thin cell membrane protrusion that we call cytonyms. So we thought that the cytonyms might be involved in the formation of clumps. And these clumps in cell culture, the formation is concentration dependent. They are generally formed in cell culture when the parasite reach 10 to the 6 parasite per mil. And if we quantify the number of cytonyms in the parasite, we also observe that there is an increase in the number of cytonyms at the concentration where the parasite formed the clamps. So we think that the cytonins might be contribution to the formation of these cell clamps in cell culture. But also what we observe is that these cytonins are generally formed connecting two parasites. Here I'm showing you by electron microscopy two parasites, and they seem to be connected by this cytonin. So we thought that these cytonins might have a role in parasite-parasite communication. And why do we think this is important? In 2014, the Shane Carton group, they performed a work where they isolate 211 samples from patients all over the world, and they found that 11% of the samples were mixed infection. What does it means that the patient was infected by more than one strain. And this is not surprising because I told you before that this parasite is usually asymptomatic, so it's not treated, the infection remains chronic, and if we consider the prevalence of the disease, it's expected that the patient could be infected by more than one strain. So in this situation, we thought, okay, what happens if the patient is infected by two different strains? Is it possible that these strains interact and one strain could be affected the behavior of the other strains in the patient? So to answer this question, what we first did is to stain one of the strains that it's highly adherent and forms a lot of cytonins and philopodia, that is called CDC 1132. We stain CDC 1132 in green, and then we stain in red 3 different strains, CDC 1132 as a control, G3, and B7RC2. And we first evaluate if CDC 1132 is able to form the clamp with the different strains, and indeed, they form the clamps. And then we analyze the number of cytonins that this parasite have in the presence of another strains. And what we observe is that if we mix CDC 1132 with a different strains, that is here G3 or B7RC2, the parasite form more cytonins than if we mix the parasite with itself. So the formation of cytonins seems to be affected by the presence of a different parasite strain. So then we wonder if this is an effect that is contact dependent. And to evaluate this, we use a transwell system that has a 1 micrometer pore filter that allows the passage of secreted factors as well as extracellular vesicles. And we put on the bottom of the transwell CDC 1132, and on the top of the chamber, we put CDC 1132 or two other different, or G3 and B7RC2 that are different strains. And then we quantify the number of cytonin that CDC 1132 is forming. And what we observe is that if we put on the transwell G3 or B7RC2, the number of parasites containing cytonin on the bottom of the well is higher than if we put CDC 1132 on the top. So there is a specific response suggesting that there is a specific secreted factor that is inducing the formation of this cytonin. And if we think about secreted factors and communication, the stars are the extracellular vesicles. So extracellular vesicles are nano to micro-sized particles that are released by any type of cells and cannot replicate by their own. And they are considered the major mediator of communication in most of the cells. There are different types of extracellular vesicles that depends on the size and the biogenesis process, but the most studied are the exosomes. The exosomes are small size extracellular vesicles that are formed inside the multivesicular body as intraluminar vesicles. And when these vesicles reach to the surface of the cells, they are released as exosomes. And on the other side, there are a different type of vesicle called microvesicles or shedding vesicles that these vesicles protrude from the plasma membrane. They are bigger and they have different markers. So in our lab, we have been working with extracellular vesicles for a while, and we developed different protocols to specifically isolate exosomes or microvesicles. Here I'm showing you a laser scattering analysis where you can see the size of the population from the vesicles that we are isolating. So these samples are enriched in exosomes because are small, and these samples are probably enriched in microvesicles because they are bigger. So we developed these protocols, and we analyzed the proteome of these vesicles, and we observed that these vesicles are different. They have different protein content, although they share a lot of proteins between them. And then we analyzed some function of these vesicles. We demonstrate that there are virulence cargo sorting in these vesicles. They have a role in attachment. They have immunomodulation properties. And what is important for this talk is that in our previous work, we demonstrate that BPS32 has a role modulation extracellular vesicle biogenesis. We demonstrate that if we overexpress BPS32, that is a member of the SCAR complex and is involved in this cell membrane scission, we observe that if we overexpress BPS32 and we isolate extracellular vesicles from the same number of parasites, we consistently found that the parasite that overexpress BPS32 that I'm showing you here by electron microscopy and by NTA analysis, they produce more vesicles. So we know that BPS32 produce more vesicles, so we use this as a tool to evaluate if the vesicles are involved in the formation of cytonims in recipient cells. So what we did is to use a similar scheme as the one that I showed you before, but now we include BPS32 and an EP-neo, that is our control, that is transfected with an empty plasmid. And then we put BPS32 EP-neo on the top of the well, G3, that is the strain that is transfected with BPS32, and CDC 1132. And we quantify the number of cytonims of CDC 1132 on the bottom of the well. And we know that these overexpress extracellular vesicles. So if the extracellular vesicles are responsible of the formation of cytonims, we are going to have more parasite with cytonims in this well. And this indeed is what happening. If we quantify the number of parasites containing cytonin, we see that the ones that are in contact with the BPS32 overexpressing parasite for more cytonin, suggesting that the extracellular vesicles are responsible, at least in part, of the formation of cytonins. And to validate this, what we did is to isolate vesicles from these 3 parasite strains to look at and then we expose CDC 1132 to these isolated vesicles. We quantify the number of cytonins on Cephalopodia, and indeed we observe that if we incubate the parasite with vesicles that were isolated from G3 or B7RC2, the parasite have more Philopodia and cytonins than if we incubate with vesicles that are coming from the same strain. So there are some protein or some factor that is inducing the formation of these cytonins. And to evaluate this, we perform a proteomic analysis trying with the aim of identifying the proteins responsible of this response. We isolate 3 independent extracellular vesicle population from the 3 different strains, and what we found is that we consistently identified a huge different number of protein in each of the vesicles isolated. If we isolate vesicles from B7RC2, we consistently found around 400 proteins, where if we isolate vesicles from G3, we have around 1,400 of proteins. So the number of proteins that we identified were completely different. Here, I'm showing you a heat map to have an idea of how different these vesicles are. So this was a little bit disappointed for us because we were expecting to have like 5-10 different proteins, and these were going to be the responsible of the effect in communication that we were looking at, and it was not the case. The vesicles itself seems to be completely different. And this is probably, I think it's an important take-home message if we are working with pathogens, that if we have one result in one strain, we need to have in mind that in a different strains, the things could be completely different. So the vesicles itself seems to be completely different. However, if we analyze by Go term the proteins that are contained within these vesicles, it was surprising to see that the biological process and the molecular functions were absolutely conserved. We were surprised to see how conserved the function of the vesicles seems to be. So the vesicles seems to be different, but the function seems to be highly conserved. So we know that the vesicles induce the formation of cytonins, and these cytonins in other cells have been demonstrated to have a role in adherence. So we decided to evaluate the formation of cytonins in contact with host cells, and what we found is that in a lot of cases, we found parasite attached to host cells that seems to be attached through the formation of these cytonins. And here I'm showing you a video of parasite exposed to host cells, and the parasite seems to be attached through the formation of these cytonins and also the host cell seems to be recognizing the cytonin and changing the direction of this cell protrusion. So if we consider that the extracellular vesicles induce the formation of these cytonins, and these cytonins could have a role in attachment, what we did before is to evaluate if the communication between these different strains could be affecting the attachment properties of the parasite. And to this end, what we did is to put CDC1132 in the bottom of the well. But now, instead of quantifying cytonins, what we did is to grow cells on the bottom of the well and to measure attachment. So we measure attachment of CDC1132 in the presence of different strains in the top of the transwell. And what we observe is that if we put on the transwell a different strains, CDC1132 attach better to host cells than if we put the same strains, suggesting that the strain is influenced by the presence of the other strain. However, CDC is already a very highly adherent strain. And we wonder what happens if a patient is infected by a highly adherent strain. What happened in a poorly adherent strain? Is it possible that the highly adherent strain affect the behavior of the poorly adherent strain? So to evaluate this, we did the similar experiment, but we switched the strains on the transwell. And now we put a poorly adherent strains on the bottom of the well and a highly adherent strains on the top. And we evaluate attachment of the G3 strain to prostate cells. And we observe that indeed the poorly adherent strains have increased the attachment if the highly adherent strain is present, suggesting that the communication between strains could be affecting the behavior. And this could be important when the patient is infected by more than one strain. So this could have an effect in mixed infection. So as a summary from this part, I show you that different strains could communicate by the release of extracellular vesicles, that these vesicles are able to form cytonins, and these cytonins might have a role in parasite-parasite communication and clamp formation, as well as a role in adherence. So I told you that cell protrusions are emerged as a way to communicate between cells, but most of the research done in cell protrusions happen on the anterior part of the cells. But the posterior part of the cells are also important because the cells, this is not only important for communication, but also for migration. The cells need to polarize and it attached the surface to allow the movement. So when we were doing this work in cytonins, we observed that the parasites that are highly adherent to host cells, they also have a huge cell membrane posterior protrusion on the posterior part of the cell that is not present in the poorly adherent strains. Here on the top, you have a G3 strain that is poorly adherent. And if you look at the posterior part of the cell protrusion is minimal, where the highly adherent strains have a huge cell membrane protrusion. So we wonder, what is the role of this cell membrane protrusion in the pathogenesis of these parasites? So we quantify the posterior cell membrane protrusion that we call uropod in 3 different strains, one that is very highly adherent in blue, a medium adherent in pink, and a poorly adherent in orange. And we observed that the highly adherent strains has a lot of cell membrane posterior protrusion, this uropod-like structure. So we thought that might have a role in attachment. So we exposed the parasite to host cells, and we observed that the parasites are usually attached to the host through the formation of this posterior protrusion. Here I'm showing you a video of a parasite exposed to host cells. And what we observe is that here you have a prostate cells and a parasite that is attaching to the prostate cells and seems to have this posterior part, cell membrane protrusion, seems to have a role as an anchor structure through the host cells. So and also in mammalian cells, the formation of self-protrusion has been modulated by proteins that are being called tetraspanine. Tetraspanines are proteins that are transmembrane domain protein that has a role in cell motility, in adhesion, migration, and also in the formation of this cell membrane protrusion. In our lab, we have been working with tetraspanine for a while, and we have parasites that overexpress 5 different cell membrane tetraspanine that we call tetraspanine 1, 2, 5, 6, and 8. And we thought that maybe this tetraspanine might have a role in the formation of this cell protrusion as well. So we quantified the parasite that overexpress each of these tetraspanine. We quantified the number of posterior cell membrane protrusion, and we found that the parasite that overexpressed tetraspanine 5 have a lot of cell membrane protrusion that if we look in detail in electron microscopy, we observe that we quantify 4 times more parasite containing cell membrane posterior protrusion than the control that is transfected with empty plasmid. And if we look at by electron microscopy, these protrusions are very thin and they seem to be much bigger than the control. So we think that the tetraspanine 5 have a role in the formation of this cell protrusion. And as the cell protrusions, the uropod cell protrusion seems to have a role in attachment, we transfect 2 different strains with tetraspanine 5, and we evaluate the capacity of attachment of this parasite to host cells. To do this, we stain the parasites with cell tracker blue, and we attach the parasite to the monolayer, and then we quantify the number of parasites attached to host cells. And here I'm showing you a picture, representative picture of the results. But in both cases, we observe that if we overexpress parasite with tetraspanine 5, the attachment of this parasite is increased, suggesting that the formation of this posterior cell membrane protrusion might have a role in the increased attachment of this parasite. We did the same and look at the by electron microscopy, and again we see more parasite attached to host cells, and in several cases the parasite seems to be attached through the formation of this uropod-like protrusion. And this is not only host cells. If we put the parasites alone to grow in a fibronectin coat slide, we also see that the parasite seems to be attached to this protrusion and working as a tethering structure that allows the parasite not to move and could be influenced migration as well. we thought that if the parasite is anchored to this structure, the migration capacity could be also affected. So we inoculate the parasite in semi-solid agar plates as people in Trypanosoma brucei does to evaluate social motility. And we inoculate the parasite and allow the parasite to grow for 5 days. And what we observe is that parasites that overexpress in TSP5 that have this huge uropod-like protrusion has reduced capacity of migration in agar plates. If we quantify this, we see that in both strains transfected with the transpanin 5, the parasite has less capacity of migration, suggesting that this cell membrane protrusion that we now identify could have a role as an anchoring structure that might have affected the parasite migration as well as adherence to host cells. So we identify a tethering structure, a novel tethering structure that might have a role in pathogenesis, and now we are trying to knock out the gene to see if this is the case. So as finally, I want to thank my group, especially Newen and Manuela, who were in charge of this project, and the funding agency and our collaborators. So, thank you.
Hello, Natalia, can you hear me?
Yes, now I can.
Thank you so much for a wonderful talk. So as always, I'll open it up for questions, and those online could just raise their hands and I can unmute you if you have a question. Maybe I'll selfishly go with the first question. That looked amazing, the way the kind of communicate with each other. I wish my malaria parasites could hold hands and communicate. I was just wondering, so basically, if you have multiple strains, that basically gives them an advantage with both addition to host cells and to migration. So why do you think that is? Like, why do you think, does this have any evolutionary implication to have like a mixture?
We actually don't know if this is an advantage or because the cells, the presence of more than one strain could be an advantage or could be a competition as well. We still don't know if this is good for the parasite or not. We are now starting to evaluate the resistance to the antibiotic in the presence of these strains. What happened if we put a highly resistant strain to metronidazole and a poorly adherent strain and a sensitive strain, maybe they have this communication could have a role in drug resistance as well, we think. But we still don't know if this communication that happened could be beneficial or a competition.
Thank you. Richard?
Natalia, the cytonides that you described, they're amazing looking. What are they composed of and how do they form from the cell? Do you know?
We know that they have actin. Are you working for are you asking for the composition?
I'm wondering if there's just an extrusion of the cell membrane or there's some specialist structure.
We think that is an structure, an extension of the cell membrane. We know that it is composed by actin, but we don't know what else is in there.
Are they big enough to allow another form of communication between the cells?
We still don't know.
Okay.
Well, actually, one of my collaborators, I work with another lab from Brazil, and he's looking at he thinks that there is a double membrane, which is weird. So we still don't know if this is truly an extension of the plasma membrane, because in his last email say, "Oh, I'm looking at a double membrane," which is Or maybe it's a vesicle that is pushing.
Yeah.
We don't know
Okay.
yet.
Thank you.
I'm going to alternate between virtual because there's a lot of people online too. Claire, if you could, I have unmuted you.
Thanks. Thank you so much for that talk. That was beautiful. And the
Thank you.
videos are just incredible. I had a question. So it looks like the protein composition of these vesicles is really heterogeneous. Do you have any idea what the signal could be to the cell that's telling it to change its attachment properties, for instance?
Honestly, I don't know, because we didn't expect that. And for me, it was really disappointed because I wanted to have like a few proteins, but it's science, it's like that. I don't know why the vesicles are so different. This is not what I expected. But as a bad PI, I told my student, no, you did something wrong. It's not possible. This is what I thought. It's not possible that you get 400 proteins and 1,400 in the other sample. This is contaminated. You have contamination. And he did 6 different samples, independent samples, and all it's consistent. I don't know why. I don't know why. But the functions seems to be highly conserved. So the vesicles are working as a vesicles, but they are different. I honestly, I don't know why.
Have you looked at like RNA or metabolites or anything like this?
There is another group in New Zealand that is working in the RNA in vesicles, and the vesicles from Trichomonas are full of tsRNA. Then there might be a difference in tsRNA as well. I didn't look at that.
Thank you.
Hi, Natalia. Really nice talk. Just a question about the virulence of these strains, because apparently the ones that have a higher attachment, I would expect them to be more virulent in the organisms, but they seems to spread less according to the last data you showed. It's like the ones that attach more poorly are more efficiently disseminated, or could be more efficiently disseminated. Is that true? Or
I think the parasites that are highly adherent, they attach to anything. They are very adherent, and in general they are also very cytotoxic. The strains are very different. I'm not confident enough to say that the result that we get on the plate, and like the social motility and the colony growth, it correlates with the attachment to host cells. But it might be, how, as you said, that the highly adherent strains attach and they don't move. So they form this posterior cell protrusion, and it's attached, and they cannot move that much as the other one, that maybe they are more motile and they can, but I'm not sure. These strains are very, very different. In Trichomonas, we don't have a lot of tools, but so we work a lot with these strains that are very well characterized because of the lack of tools, clearly, right? And we know that these strains that are very highly adherent, they are also very cytotoxic. And when I mean highly cytotoxic, is that if I put these strains with the host cells, in 1 hour, you don't have any cells, any host cells. They destroy the host cells. And opposite, if we put the poorly adherent strains with host cells, you can leave it like 24 hours and the monolayer will be there. So the phenotypes are very different. And we work with that a lot because it allows us a comparison between the strains. But I'm not confident enough to say that this migration difference is correlate with the virulence in the host cell.
thanks.
Test online.
Test is the name, or
Maybe not. If you could.
Hello?
Hello.
Oh.
Yes, we can hear you.
Okay, hello, Natalia. It's Robert Hurd speaking. Nice talk as always.
Robert. I know him.
¿Todo bien? So, very nice talk. I just had a question. Have you considered the idea that maybe those interactions between different strains may trigger or be involved in some sexual interaction between the different strains?
Well, it's possible. I never test that. There are some, well, you know that, but there are some published work from Shane Carton group that suggests that Trichomonas could have all the
That's
genes.
exactly why I asked. Yes, yeah.
Yes.
Yes.
Trichomonas could have the genes for sexual reproduction, but there has not been work done on that. Last year, I went to the Gordon conference in Salva Regina, and there was a work with Plasmodium related to that. And I came back and said, oh, we are going to do that to evaluate if the parasite could have a sexual reproduction, because in Plasmodium, they said that their sexual reproduction is only inside the clamps. So it correlates with our clamps, maybe, I don't know. But I haven't tested, honestly.
Okay, that would be nice to look into. Yeah, thank you very much.
Yeah, I agree.
Hi. I would like to come back for a moment to the proteomic composition of the vesicles. Those proteins, are they the same proteins that are found in cytoplasm, or are they excretory only? Do they have a particular signal sequence, perhaps? So how would you classify them in the rest of the cellular biology of Trichomonas?
I'm not sure if I listen your question correctly. Did you ask if the difference in the protein composition is specifically in one pathway or something like that?
Are these secretory proteins? Yeah, they have maybe a signal sequence to be secreted.
We have like 800 protein difference. It's a lot. So they're everywhere. They're all different. We couldn't say, okay, the difference is in this family or this pathway, or because maybe we have, I don't know, ribosomal protein, but in one extracellular vesicle we have like 10, and in the other we have 80. So it was in all the protein families. It was not specifically in one part. We couldn't do anything.
So I wonder if this is evidence that there is just a subsection of the cytoplasm is taken into those vesicles.
You mean, no, in theory, there are some type of specificity. So the vesicles, there are some proteins that are specifically located inside the vesicles. It's not like one part of the cytoplasm randomly goes to the vesicles. Is that what you mean?
Yes.
Okay. No, it's supposed to have some specificity in the protein content. There are some in other cells. I'm not telling you in Trichomonas because we haven't tested, but in mammalian cells, there are some sorting to the vesicles. There are some signaling. That was your question. Sorry. Now I understand.
Francis, online.
Hello, can you hear me?
Yes.
Yes.
Hi, thank you. Thank you, Natalia. I have a kind of quite Trichomonas specific question, because I was in Jane Carlton's lab previously
I
working
remember.
G3 genome. And I kind of got the impression that it's a bit of a mess, the G3 strain, in comparison to other more sort of wild type strains, just because it's been in the lab for a long time. And I was interested that I think I was thinking along the same lines as Richard, that these protrusions are maybe some part of the vesicular pathway that's not getting made into vesicles, it's getting made into a long thing instead. And I noticed that's kind of broken in G3, that you don't get this posterior protrusion. And you also have that massive amount of proteins in there compared to the other strains in the
I totally agree with you. I hate G3.
Yeah,
because it's the problem is what Francis said. We all hate G3 because it's a mess and it has been in the lab forever, but it's the only genome we have. So we are always in the situation where we choose the strains that we have the genome sequence, or we choose strains that we like more because it's more physiological, or it's more like isolated from patient recently. So I use G3 for the reason that we have the genome, but I totally agree with you that the result from G3, that's why I always put another strain that is not G3 to see if I have the same results, because I don't trust a lot the result with G3 alone.
Yeah, I was going to ask if you have kind of high quality genomes for those other two strains, because they seem to have more similar numbers of proteins getting pushed into the EVs. So that would be so useful to compare between those more realistic strains, which proteins are actually going into the EVs and which part.
Yeah, I totally agree.
All that stuff.
No, we in the lab we have another project that is in epigenetics. So we work in cell communication and we work in epigenetics. And for the epigenetics project, we are now doing a lot of work in different strains, and we are trying now to sequence one of the genomes because it's a problem for us. We don't have the genome yet.
Yeah, those genomes are not straightforward as well. So, but that's awesome. I look forward to seeing how that goes. Thank you very much.